nervous system video. First, we're going to describe the three essential roles of the nervous system, and they are sensing, integrating, and responding. So, sensing, the nervous system uses sense organs to detect changes both inside and outside the body. Integrating is like the thinking. We process that information related to past experiences and determine an appropriate response. And then the response is going to be nervous system sends a command to the muscles. So, for example, you see a bear. You see teeth and claws, and you hear growling. And your past experience might tell you that teeth and claws and growling is a bad thing, so your response might be to run away. Next, we're going to look at the divisions of the nervous system because it's so complex and does so much. We divide it up into different divisions. So first, the central nervous system consists of the brain and spinal cord, and that is primarily what does the integrating. And then the peripheral nervous system is all the nerves. So in this figure, the central nervous system is in that turquoise color, and then the yellow is all the rest of the nerves, and they're doing the sensing and responding. So the peripheral nervous system can be further divided into the sensory or afferent division. Afferent means going towards. So this division carries signals from nerve endings to the CNS, which is short for central nervous system. And the sensory division can be further divided into the somatic sensory division, which carries signals from skin, bones, joints, and muscles. So all our touch information is going to go through the somatic sensory division. And then the visceral sensory division carries signals from the viscera of the heart, lungs, stomach, and bladder. So that might be pain, or it might be more things that happen automatically that we don't have to think about. Then we have the motor or efferent division. Efferent means going away or exiting. So it is transmitting information from the CNS to the rest of the body. And it can be divided into the somatic motor, which controls our skeletal muscles, our voluntary movements, and the autonomic motor, which provides automatic things. It controls our smooth muscles and cardiac muscles, so it controls blood pressure, heart rate. And then this autonomic division can be further divided into the sympathetic division, also known as the fight or flight division, and the parasympathetic division, also known as the rest and digest division. We'll talk more about the ANS at the end of the chapter. Alright, so the primary cell of the nervous system is the neuron. Those are the ones that do the communicating. The supportive cells are known as neuroglia or glial cells. And a couple of ones you need to know their function. Oligodendrocytes form the myelin sheath in the central nervous system. We'll talk about the myelin sheath in a bit. Ependymal cells line the spinal cord and cavities of the brain, and these are involved in producing cerebrospinal fluid. So the ependymal cells produce the cerebrospinal fluid. Then the microglia perform phagocytosis. So your normal immune cells that would do that generally don't go in the brain and spinal cord. So it has its own sort of immune system, which is what the microglia do. Then astrocytes form the blood-brain barrier, which we're going to talk about more in a second. That keeps harmful things out of your brain. And then Schwann cells are forming the myelin sheath in the peripheral nervous system. So here is the blood-brain barrier. It's made up of these astrocytes. They're called astrocytes because they sort of look like stars, and they have these foot processes that go out and surround the capillary. So on the bottom you can see a normal blood vessel, and then one in the brain where it's surrounded by these foot processes from the astrocytes, and it protects the brain from harmful things. A lot of drugs can't get through that. Alcohol can go right through it, so it doesn't protect us against that, but it does protect us from some drugs, and it also keeps like bacteria out of the brain. It also keeps our own immune cells out of the brain, which is why we have the microglia inside the brain. Then we need to know what type of cell is likely to form a tumor. So neurons, once the brain is formed, we really don't want the neurons to change, so they don't divide. And cancer is when cells start dividing out of control. So if you're not dividing at all, you're not very likely to form cancer. The glial cells do divide, so they're much more likely to form a tumor. They need to be treated by chemotherapy, or sorry, not chemotherapy. Chemotherapy doesn't work because of the blood-brain barrier. Surgery and radiation are the treatments. There are ways to get around that. We can put chemotherapy wafers directly in the brain, but that's a pretty invasive process. Not near as easy as hooking up an IV. Alright, so neurons again are the primary cell. They handle communication. We need to know their classes. Basically, they are sensory, which are detecting stimuli and sending it to the CNS. Interneurons are connecting pathways. Those are going to be found in the CNS, in the brain and spinal cord. And then the motor or efferent neurons are going to be relaying messages. Now we need to know the structure of a neuron. So the cell body is going to be the largest part, and if you look at your hand is a good neuron model. The palm of your hand is like the cell body. It's going to contain the nucleus, mitochondria, ribosomes. It's not going to contain centrioles because they don't divide, but it has all your usual organelles, and it's going to be, one of its jobs is making neurotransmitters that will be released from the end of the neuron. Then the dendrites sort of look like tree branches. They're out around the cell body, so if you hold out your hand and hold out your fingers are like the dendrites. They're like the input part of the cell. So other neurons would be sending messages to the dendrites, and then they relay it to the cell body. And then the axon, so your arm would be the axon. That's the part that's leaving the neuron, so that's the output part of the neuron. It's sending signals away from the cell body on to the next neuron, or possibly to a muscle or gland. Then this axon has a myelin sheath. Those purple hot dog shaped cells make up the myelin sheath, and its job is to it protects axons. It also speeds up action potentials, or the electrical signals that are carried along the axon. The nodes of Ranvier are the spaces in between these cells. These are Schwann cells making up the myelin sheath here, and the action potential normally has to be generated all along the axon, but with the myelin sheath it just has to be generated at those nodes of Ranvier, so that allows the action potential to travel much faster. Then the synaptic knob is at the end of the neuron, and it is going to release neurotransmitters. Usually the axon is going to branch, and it can contact multiple other neurons or muscle cells. Alright, next is the structure and function of myelin sheaths. So on the left you can see Schwann cells in the peripheral nervous system. On the right, oligodendrocytes in the central nervous system. Oligodendrocytes can surround more than one axon, which is handy in the brain where there's axons all over the place. The Schwann cells in the peripheral nervous system are going to be forming just wrapping around one cell. And as they wrap around the cell, the cytoplasm gets squeezed out to the edge, and you're left with a layer of basically just the plasma membrane, which, remember, is composed primarily of phospholipids, which are mainly fat. So basically it's a layer of fat around your axons, but fat is a really good insulator, so it works really good, and again it speeds up the action potentials. Alright, next question is why a child younger than 2 should never be placed on a diet. So this process of myelination, or forming the myelin sheath, is not complete until late adolescence, and it's going on especially during the first 2 years of life. And since that myelin is mostly fat, you need fat in order to make that myelin sheath. Alright, next thing is action potentials. So a question that which neurons detect sensations and relay information about stimuli to the central nervous system? And the correct answer is the afferent neurons. They're going to be sending information towards the central nervous system. Alright, we're going to watch this animation.
To relay messages to organs and tissues throughout the body, nerves must initiate and then transmit signals from one neuron to the next neuron at lightning speed. Signal transmission occurs through an electrical current. When a neuron is not conducting an electrical signal, its interior has a negative electrical charge, whereas the charge on the outside is positive. The outside of the cell is rich with sodium ions, whereas the inside contains an abundance of potassium ions. The interior of the cell also contains large, negatively charged proteins and nucleic acids. These additional particles give the cell's interior its overall negative charge. Because of the membrane's permeability, a certain amount of sodium and potassium ions leak across the membrane. However, the sodium-potassium pump constantly works to restore the ions to the appropriate side. This state of being inactive and polarized is called resting potential. The neuron is resting, but it has the potential to react if a stimulus comes along. When a stimulus comes along, channels on the resting neuron's membrane open, and the sodium from outside the membrane rushes into the cell. The addition of all these positively charged ions changes the charge of a region of the cell's interior from negative to positive. As the membrane becomes more positive, it is said to depolarize. If the stimulus is strong enough, adjacent channels also open, allowing even more sodium to flood the cell's interior. This creates an action potential, meaning that the neuron has become active as it conducts an impulse along the axon. The sudden influx of sodium triggers the opening of other channels to allow potassium to flow out of the cell. Soon after potassium begins to exit, the sodium channels shut to prevent any more sodium from flowing into the cell. This repolarizes the cell. However, the outside now contains more potassium, and the inside contains more sodium. Although the membrane is polarized, the neuron will not respond to a new stimulus as long as the sodium and potassium are on the wrong sides of the membrane. The sodium-potassium pump works to return sodium to the outside and potassium to the inside. This is known as the refractory period. When the sodium-potassium pump restores sodium ions to the outside of the cell and potassium ions to the inside of the cell, the nerve is again polarized. It remains in resting potential until it receives another stimulus.
Alright, so neurons communicate with other cells using action potentials or nerve impulses. So this impulse conduction is caused by an electric current. Membrane potential is when ions with opposite electrical charges are separated by a membrane. The plasma membrane is a pretty good barrier for charges, so we have this membrane potential. And polarization is when a membrane has an excess of positive ions on one side and an excess of negative ions on the other, and that is the case in our neurons. So normally they have a negative charge on the inside when they're at rest. Something happens, a neurotransmitter binds, and that opens some channels and allows sodium to enter. As these positive sodium ions enter, the cell becomes more positive, and then it spreads like dominoes. Once you open one channel, it's going to open the next channel. Those positive ions will cause more channels to open and more sodium to come in, and it's just like dominoes. And that's how the action potential spreads down the neuron. Then following that, we have repolarization, where the potassium is leaving the cell, and as those positive potassium ions leave the cell becomes negative again, And then we get in this refractory period where the sodium potassium pump is pumping the sodium out and the potassium in and getting it reset and ready to send another action potential. So another term for nerve impulse is action potential. Alright, multiple sclerosis is caused by the myelin sheath deteriorating. Symptoms include visual disturbances such as blindness or double vision, muscle weakness, loss of coordination, loss of the ability to speak. It was thought to be an autoimmune disease. Now there's more and more evidence that it may be a microbiome imbalance causing this. Alright, next thing is how neurons communicate with each other at a synapse. So let's watch the animation.
Nerve impulses usually travel through several different neurons before reaching their target organ or tissue. For this to happen, the impulse must have some way of transferring from the axon of one neuron to the dendrite of another neuron. The area where this occurs is called a synapse. Most synapses do not touch. Instead, a chemical called a neurotransmitter bridges a very narrow gap called the synaptic cleft to carry the message from the first neuron, called the presynaptic neuron, to the next, called the postsynaptic neuron. The process begins when an action potential reaches a synaptic knob and the membrane depolarizes. This causes ion channels to open, which allows calcium ions to enter the cell. The infusion of calcium causes vesicles to release their store of a neurotransmitter, such as acetylcholine, into the synapse. Once released, the neurotransmitter binds to receptors on the postsynaptic membrane. This causes sodium channels to open, and sodium diffuses into the cell. The addition of the positively charged cells causes the membrane to depolarize, and the impulse continues. The receptor then releases the neurotransmitter, after which it is reabsorbed by the synaptic knobs and recycled or destroyed by enzymes.
Okay, so to review, at the synapse, an action potential is going to reach the end. That causes calcium to enter. Then the next step, these vesicles are going to release the neurotransmitter. That's what these little blue balls are. Then the third step, the neurotransmitter is going to bind to the receptor. And then the fourth step, the neurotransmitter determines if the impulse will continue. In this case, it's an excitatory neurotransmitter, and sodium enters, and we get a new action potential in this next cell. A different neurotransmitter might cause it to stop. And then the very last step, the receptor is going to release the neurotransmitter, which can then be recycled or broken down. And because we know about synapses, we can make drugs like Prozac, one of the most commonly prescribed drugs in our country. It's known as a selective serotonin reuptake inhibitor, or SSRI. And so normally serotonin gets reuptaken into the cell that released it. And what Prozac does is it blocks those channels that take the serotonin back into this cell. So that way it sort of tricks your body into thinking that you're releasing more serotonin than you are, because the serotonin ends up staying in the synapse longer. And it helps a lot of people with depression. Alright, next is the function of the spinal cord. It is an information passageway. Sensory information goes through it to the brain, motor information goes through it from the brain, and it also is responsible for reflexes, which we'll talk about in a bit. We need to explain why lumbar punctures are performed between L3 and L4. So, before you're born, when you're in your mother's uterus, your spinal cord goes all the way down to the end of the sacral vertebrae. But as you grow, your bones grow faster than your spinal cord grows, and by the time you're born, the spinal cord is about the level of the third or fourth lumbar vertebrae. And by the time you're an adult, it's about the level of the first spinal cord. So gray matter is found in the center of the spinal cord. Sorry, this figure, I guess it sort of looks gray in this figure. It contains cell bodies and interneurons. And then around the outside you have the white matter. White matter is white because it's myelinated axons. Remember, the myelin is mostly fat, so it looks white. And the white matter is sending signals up and down the spinal cord. The epidural space is just outside the spinal cord. It's filled with mostly fat, and if you ever get an epidural, that's where they stick a needle in there, and then they leave a little plastic tube in there to give the drugs, and then it will block the sensory receptors. The central canal is in the center, and it will be filled with cerebrospinal fluid. The dorsal or posterior nerve root is on this figure, and on the dorsal root you can see there's an arrow going towards the spinal cord, so that's where you're going to have sensory information. And then for the anterior or ventral root, the arrow is going away from the spinal cord, so that's going to contain motor information. And then the dorsal root and the ventral root join together to form the spinal nerve. There are 31 pairs of spinal nerves, one leaves between each vertebrae. And the way I remember that the dorsal root is sensory and the ventral root is motor is because if you were being chased by a bear, he would cut through the back of you, he'd reach out his claw, and he would cut through first your dorsal or posterior root. And that's okay because that's just sensory information, so now you can't feel the pain. And your ventral root is intact, that's your motor, so you can still run away. So your body is not really designed to withstand bear attacks. Please don't play with bears, but that's how I remember. Dorsal is sensory, dorsal root is sensory, ventral root is motor. Alright, next is the layers of the meninges. So the meninges surround and protect the brain and spinal cord. The innermost layer is the pia mater. It clings to the brain and spinal cord. It has blood vessels. Then outside of that you have the subarachnoid space. This is where you will find the cerebrospinal fluid, which cushions and nourishes the brain and spinal cord. Then the middle layer is the arachnoid matter, and it sort of looks like spider legs. That's how it got its name. And then the outer layer is the dura mater. It's a tough connective tissue. It's white. And those are the meninges. Now let's look if we had a spinal cord injury between T1 and L1. So the top vertebrae are the cervical, the yellow ones are thoracic, the purple ones are lumbar. So if it's anywhere basically in the thoracic vertebrae, you're going to have paralysis of the trunk and the legs, so you would be a paraplegic. If it's above C5, then you're going to have paralysis of your arms and hands as well, so then you're a quadriplegic. And if it's above C4, it's going to be complete paralysis below the neck, and that may damage your respiratory muscles, so you would have to be on a ventilator your whole life and not be able to move anything. So try not to damage your spinal cord. Alright, next thing is reflexes, so let's watch this animation.
Reflexes are a quick, involuntary, predictable response to a stimulus. Reflexes use a neural circuit called a reflex arc, which bypasses regions of the brain where conscious decisions are made. Somatic reflexes involve the contraction of a skeletal muscle after being stimulated by a somatic motor neuron. Patellar reflex is a somatic reflex commonly tested during physical examinations to identify certain diseases. Somatic receptors, located in the skin, a muscle, or a tendon, detect a sensation such as the stretching of the thigh muscle when the patellar tendon is tapped. sensory, nerve fibers send a signal directly to the spinal cord. The impulse immediately passes to a motor neuron. It bypasses regions of the brain where conscious decisions are made. The motor neuron initiates an impulse back to the muscle, causing it to contract, producing a slight kick in the lower leg.
Alright, so the steps of a reflex. First, a receptor is going to detect the sensation. There are stretch receptors in your muscles. You don't want to stretch your muscles out too much or they will break. Then the next step, in purple here we have the sensory nerve or sensory neuron, which is going to send a signal to the spinal cord. And then the third step, that signal is going to pass from the sensory neuron directly to the motor neuron. And then the fourth step, the motor neuron, which is red here, is going to send a signal out to the muscle. And then the muscle would kick. Next thing is, why is gray matter folded? So the outer surface of the brain is called the cerebral cortex, and that's where you do your thinking and memory and processing and things. And I have here up on the top left the brain of Albert Einstein, and you can see it's very wrinkly, and that's why he was so smart, because he had more surface area for his cerebral cortex. Then we have a normal brain, and then on the bottom left, that is a brain of someone who was mentally retarded, so you can see they had no wrinkles. And then we have this little meme down here. If you have a smooth brain with no wrinkles, you're cute, your brain's cute, and you can't think, so you can't be sad. But if you have a bumpy brain with gross wrinkles, it's ugly and it thinks, so it can be sad. But I'd much rather be able to think. So I'm sure you all have wrinkly brains that are very smart. Alright, this shows the meninges around the brain. You got the same three layers. The dura mater is the outer layer, then the arachnoid matter, and then the pia mater right next to the brain. And meningitis is an inflammation of the meninges. It's caused by a bacterial or a viral infection. These symptoms include fever, stiff neck, irritability, headache, drowsiness, and seizures. Viral meningitis generally goes away on its own. Bacterial meningitis, usually you end up in the hospital and need IV antibiotics. Alright, next let's watch about cerebrospinal fluid.
A clear colorless fluid called cerebrospinal fluid, CSF, fills 4 chambers in the brain called ventricles. It also fills the central canal of the spinal cord and bathes the surface of the brain and spinal cord. CSF primarily results as the choroid plexus, which is a network of blood vessels lining the floor or wall of each ventricle, separates fluid from blood. The choroid plexus in each lateral ventricle secretes CSF. The CSF flows into the third ventricle, where the choroid plexus adds more fluid. Then CSF flows into the fourth ventricle, where still more CSF is added. Some of the CSF flows through 2 tiny openings called foramina. From there, the CSF flows through the subarachnoid space, up the back of the brain, down the spinal cord, and up the front of the brain. The CSF is reabsorbed into the venous bloodstream by projections of the arachnoid major into the dural sinuses, called arachnoid villi.
Alright, so the cerebrospinal fluid, its main function is cushioning. Your brain and spinal cord are in their own little waterbed, and that protects you from most bumps on the head. If you hit your head so hard that your brain accident actually bumps against your skull, you get a bruise on your brain, and that's called a concussion. It also nourishes in this fluid. There is oxygen and glucose, and we monitor the oxygen levels of this fluid, and if it starts to get low, then we will start to breathe more. Now we're going to look at some regions of the brain. The cerebrum on top is the largest region. The cerebellum is towards the back, towards the bottom. And in the middle you have the brainstem. And we're going to look closer at the brainstem. It connects the spinal cord to the brain. So all throughout it you have a bunch of basically relay stations relaying sensory and motor information. In the midbrain you also have auditory and visual reflex centers, so the fact that you automatically turn your head when you hear a loud noise, that's a auditory reflex controlled by the midbrain. The pons is relaying information between the body and the brain, as well as the cerebellum. Then the medulla oblongata is relaying things. It also contains some very important centers: the cardiac center, which controls heart rate, the vasomotor center, which controls blood pressure, and there are also two respiratory centers in the medulla oblongata controlling your respiratory rate. So all those are very important for life. Next we have the cerebellum. The cerebellum is involved in motor functions. It keeps our movements coordinated. It receives input from sensors throughout our muscles, throughout our arms and legs, and it receives motor commands from the cerebrum as well. People with damage to the cerebellum will move very jerky, uncoordinated movements. So the cerebellum keeps our movements smooth and coordinated. In the middle of the brain we have the diencephalon, made up primarily of the thalamus and hypothalamus. So the thalamus is a gateway for sensory information. Almost all the sensory information has to go through the thalamus before it gets to the cerebral cortex. The thalamus also filters out a lot of information that we don't need. It takes things that are unimportant, things that aren't changing, and just filters them out, and we just kind of don't waste our brain power on them. Below that is the hypothalamus. It has many functions. It controls the autonomic nervous system, which we're going to talk about at the end of this chapter. It regulates things like hunger, thirst, temperature. It also controls the pituitary gland, which is located just below it, which controls basically our entire endocrine system. And it is also involved with emotion. And then all throughout the brainstem we have this light blue thing called the reticular formation, and part of that is called the reticular activating system, and its function is to keep us alert and awake. So it receives visual and auditory input, and then it sends signals to the rest of our brain telling us to wake up and stay alert. So it's nice that it's kind of named for what it does. Alright, cardiac and respiratory centers are found in the correct answer is the medulla oblongata. Now we're going to look at the largest part of the brain, the cerebrum, and it's responsible for your ability to think, remember, feel, use judgment, and move. And then some of the specific regions or lobes have specific functions. So we'll start with the frontal lobe in front. It's responsible for voluntary movements. The precentral gyrus that you see, that's responsible for voluntary movements. Memory, emotion, social judgment, decision making, reasoning, personality, these are all functions of the frontal lobe. Then on the side of the brain we have the temporal lobe. It's responsible for hearing, smell, learning and memory. And then sort of on top of the brain we have the parietal lobe. It's responsible for sensation as well as proprioception, so all our touch information is going to go to that postcentral gyrus. Then in back we have the occipital lobe. It's responsible for vision, so all our visual information goes there and gets turned into pictures. Then if we sort of pull back between the temporal lobe and the frontal lobe, we'll see the insula. It's involved in lesion. So a lesion is just damage to the brain, and a lot of what we learn about the brain is by looking at people with damage to a certain region and seeing what they can't do, because not a lot of people want to donate their brains for research. So someone with a parietal lobe lesion will ignore objects on the opposite side of their body. So the sensory information crosses in the spinal cord, so the right side of your brain receives sensory information from the left side of your body. So someone with damage to, say, their right parietal lobe might ignore their left arm and leg. With a temporal lobe lesion, they lose their ability to identify familiar objects, even their own face. They also lose the ability to differentiate between sounds, which makes music be not special anymore. They can't enjoy music. With a frontal lobe lesion, personality disorders, and socially inappropriate behavior. Alright, let's look at some more brain regions. So in green here you see the corpus callosum, which connects the two halves of the brain. You have a right and left hemisphere. So you have two of each of those lobes that we just talked about, and the corpus callosum connects the two sides. The limbic system deals with emotion. It's several parts of the brain. Two important parts of it are the hippocampus. Hippocampus converts short-term memory into long-term memory, and the amygdala, which stores and can recall emotion. Alright, next we're going to look at what you need to do to read something. So in order to read something, first, it's going to go from your eyes to the occipital lobe in the back. Then next it's going to go to the cingulate gyrus, angular gyrus, translates the written words into a form that can be spoken. Then it will travel to Wernicke's area, where it turns those words into phrases. And then next it will go to Broca's area, which plans the muscle movements required for speech. And then finally it goes to the primary motor cortex. That's going to send impulses to the muscles like our tongue and our lips that we need to actually say the word. And so there are a couple of aphasias we need to know. Wernicke's aphasia. People with this, they can't comprehend what others are saying. They also have trouble forming sentences. And then people with Broca's area, they can understand speech, but they can't form words. For them it's difficult or even impossible to speak. So Wernicke's aphasia, you can't understand words. Broca's area, you can't form words. Alright, hope I said that right. Alright, limbic system is charged with, and correct answer is emotion and learning. Alright, next we're going to talk about the autonomic nervous system. It regulates activities that maintain homeostasis. It sends impulses to cardiac and smooth muscle, so it's going to control things like our heart rate, our breathing rate, digestion. It's also called the visceral motor system. And if we look at an autonomic versus somatic pathway, so here's a somatic pathway. There's one neuron that goes from the spinal cord out to the muscle. Whereas with the autonomic pathway, there are two motor neurons, and in between them there's what's called a ganglion. So we're going to look at the two different divisions. The sympathetic prepares the body for activity. Again, it's known as the fight or flight. The parasympathetic calms the body down and conserves energy. It's known as the rest and digest division. So this figure shows the sympathetic on the left and the parasympathetic on the right. So the sympathetic originates sort of from the middle of the spinal cord, from what's called the thoracolumbar region. And then the parasympathetic originates from the cranial and sacral regions of the spinal cord. location of the ganglia. With the sympathetic, the ganglia are right next to the spinal cord. And with the parasympathetic, the ganglia are out near the organs. And then the neurotransmitter used, this table will help us. Sympathetic uses norepinephrine, whereas the parasympathetic uses acetylcholine. And then the sympathetic produces widespread generalized effects, whereas the parasympathetic produces more local effects. And we need to know why the sympathetic division can produce an immediate widespread response, and that's because it's preganglionic fibers here. They can synapse on more than one postganglionic fiber, so that amplifies the response. And then we need to explain dual innervation. So you can see most things are innervated by both the sympathetic and the parasympathetic. So like your heart and lungs, pretty much every organ is innervated by both. And so for example, when you're running a race, your sympathetic is going to be dominant, and that's going to speed up your heart rate. Right now, while you're sitting here watching this video, probably not feeling threatened, your parasympathetic is going to be dominant and your heart rate is going to be low. So dual innervation means that most organs are innervated by both sympathetic and parasympathetic, but depending on the situation you're in, one or the other will